Jayambe, how are you all? I hope you all are good. I welcome you all to my YouTube channel. My name is Prince Ranpara. I am a PhD scholar in the Department of Chemical Engineering at the Indian Institute of Technology, ROPO. So in today's lecture, we are going to understand the batch reactors, right? So in today's lecture, we'll understand how we are implementing the concept of the general mole balance equations in order to derive the design equation of the batch reactors, right? So let's get started, right? So first of all, in our mind, the question arises is what is the batch reactors? How the batch reactors differs from the industrial reactors, right? From the other industrial reactors, how the batch reactor differs. The third question which arises in our mind is what are the industrial practical applications of the batch reactors, right? So in today's lecture, we'll understand and you will get the answers of all the questions, right? So let's get started. So first of all, we can define the batch reactor as the batch reactor can be defined as the closed vessel. Okay, so batch reactor can be the batch reactor, which means the closed vessel in which the reactants are fed in to the reactor, right? The reactants are charged into the reactor. Okay, so in the reaction environment, in where the reactants are charged, right? So here in the reaction environment, we'll have the presence of the reactants, right? And we'll also have the presence of, if we are using any solvent, then in the reaction environment, we'll have the presence of reactants or solvent, or if we are using any catalyst in our reactions, right? Then the presence of reactants, solvent or catalyst in the reaction environment. Okay, so in the reaction environment where the reaction takes place, right? And during the progress of the reaction, during the run of the reaction, there will be no inlet flow rate and there will be no outlet flow rate. Okay, it's termed as the batch reactor, which means a batch where the reaction will takes place, right? So in order to understand the batch reactor, where we have the volume of the reactor in which in the reactor system we feed in the reactants, right? Reactants may consist of any reactant species, or it may also consist the solvents, or it may also consist the catalyst, right? So the reactants or the solvent and the catalyst are placed inside the reaction volume, in the system volume where the reaction takes place. In order to maintain the uniform distribution of the reactants, in order to maintain the uniform distribution of temperature, concentration of species, the catalytic activity, we are using the agitator or stirrer, right? We are using the stirrer or agitator in order to provide the mixing, thoroughly mixing inside the reaction environment, due to which we'll have the uniform distribution of concentration of reactants, solvent and catalyst throughout the entire reactor volume, right? So during the run of the reaction, the inlet, there will be no inlet flow rate and there will be no outlet flow rate, right? And the reaction will take place. Okay, after completion of the reactions, the reactants will get consumed and they will react, they will consume, and we'll get the new products are generated, right? The new product which is generated is taken out from the, is removed from the reactor by the outlet stream, right? But during the run of the reaction, there will be no inlet flow rate and there will be no outlet flow rate, right? It's called the batch reactor, right? So we have so where the batch, why we are using the batch reactors, and how we, okay, what are the batch reactors, we understand, and how it differs from the other industrial reactors. Let's say in other industrial reactors like continuous starting reactor, plug flow reactor or packed bed reactors, we have the continuously feeding the reactants, right? Which means the reactants are continuously fed to the inlet stream. From the inlet stream of the reactor, the reactants are continuously feeding to the reactor, right? But in the case of batch reactor, once the reactants are fed in the reactor, no further reactants will be charged during the progress of the reaction, no reactants will be charged into the reactor. Okay, so there will be no inlet flow rate and no outlet flow rate. Okay, and reaction will takes place. Okay, so so let's say by utilization of the general mole balance concept, and we'll derive the design equation of the batch reactor, right? So when we in the previous lecture we understand the universal equation, which is the general mole balance equations. Okay, we have the Fj naught minus Fj plus integration of Rj into dV throughout the entire volume V is equal to dnj by dt, right? So Fj naught indicates the inlet molar flow rate, Fj represent the outlet molar flow rate, and the Rj into dV will represent the integration of Rj into dV represent the generation term, right? Is equal to the dnj by dt, which indicates the accumulation term, right? So now let's derive the design equation of the batch reactors, right? So let's say for the batch reactor, we understand that during the progress of the reaction, there will be no inlet flow and no outlet flow, right, during the reactions. So for those reasons, the Fj naught is equal to Fj is equal to 0 during the run of the reactions, right? The inlet term and the outlet term will be equalized to 0, because during the progress of the reaction, there will be no inlet flow rate and there will be no outlet flow rate. Okay, then from the equation 2 of the mole balance, we remain with the two term, which is the generation term and accumulation term, right? So we can write in the further equation as integration of Rj into dV is equal to dnj by dt, right? Integration of Rj into dV is equal to dnj by dt. Now let's say the reaction mixture is perfectly mixed, which means by using the stirrer, by using the agitator, we are thoroughly mixing the entire reactor volume, entire reaction volume, right? So due to which we'll have the uniform distribution of all the system variables. The system variables, right? The temperature of the system, okay, which means we'll have the uniform distribution of the temperature throughout each and every position of the reaction reaction environment in the reaction volume, right? In the system volume, means we'll have the uniform distribution of the temperature throughout the entire system volume. We have the uniform distribution of concentration of species throughout the entire system volume, and we have the uniform distribution of any reactants, solvents or catalysts throughout the entire reaction volume. Okay, due to which the rate of reaction will be uniform throughout the entire system volume. Okay, due to the uniformly distribution of all the system variables, we'll have the uniform rate of reaction throughout the entire system volume, which means at each and every position in time, rate of reaction will be same. Okay, so due to those reasons, the rate Rj term can be taken out of the integral. I hope you understood, right? So likewise, we can the rate due to the reaction mixture is perfectly mixed. The rate of reaction is the function, we already understand in the previous lectures, right? Where the rate of reaction is the function of temperature, pressure, concentration of species, or if we are using the catalyst into our system, right? In the catalyst, and the catalyst or the catalytic activities will be a function of rate of the reactions, right? Okay, which means the rate of the reaction is the function of temperature, pressure, concentration, and the catalytic activities, right? So if we are having the uniformly distribution of all the system variables, then the rate of reaction will be uniform throughout the entire system volume. Okay, due to which we can take out the Rj term from the integration, right? So Rj we are taking out from the integration, right? So Rj into dV is equal to dnj by dt. I hope you have understood how we are trying to write this equation from the above equation, right? So let's say we already know mathematical relation, right, where the integration of dV can be written as V, right? So Rj into V is equal to dnj by dt, right? So these equation represent the differential mole balance of the batch reactor, right? So this equation represent the differential mole balance over the batch reactor, right? So now let's say up to here it is clear, right? So now this equation, when we can write this equation, this form of equation, when we are having the condition of perfect mixing, means uniformly mixing of all the system variable throughout the entire reactor volume, okay, then we will be derived with the differential form of the mole balance equation, which is been indicated by Rj into V is equal to dnj by dt, okay. Now let's say if let's say in the batch reactor, in the reaction environment where the reaction takes place, right? So reaction is, let's say for example, A gives B, which means A is the reactant and B is the product. During the run of the reaction, during the progress of the time, the reactants will get consumed and will get converted into the products, right? So A will get consumed, A will disappear from the reaction environment and will get converted into new products, which is B, right? So now let's say we can understand, now batch reactors are used to understand how the concentration changes with respect to time, okay. So let's say if I want to analyze the how the moles of reactants are getting changed with respect to the time, okay. So by understanding this figure, we can understand the mole time trajectories, right? Which means how the number of moles of any reactant molecules are getting changed with respect to the progress of the time, with the progress of the reaction, and how new products are getting generated in the reaction environment, okay. So let's say we'll understand the first figure, right? So in the both the figures, where we have on the x-axis the time, right? In the both the figure, we have the, on the x-axis we have time, and on the y-axis we have the Na. Na represents the number of moles of reactants, right? And here Nb, so Nb represents the number of moles of products, right? So let's say let me make very simplified, okay. So let's say at time t is equal to 0, at time t equal to 0, now let's say let me make very simplified, at time t is equal to 0, which means at time t is equal to 0, we are feeding the reactant into the reactor volume, right? So at time t is equal to 0, we have the maximum concentration of reactants present inside the reactor reaction volume, right? So at time t is equal to 0, we have the Na0. Na0 means the initial amount of A present in the reactor reaction environment, right? So which means at time t is equal to 0, the reaction is not progress, right? So due to which we have the higher concentration of reactants, right? So for your, in order to make it very clear, simplify, let's say at time t is equal to 0, we have the 100 molecules of A present in the reaction environment, okay. Now we'll understand the nature of the curve, right? By nature of the curve, okay, nature of the curve we'll understand. So let's say in this figure, the slope of the curve is dna by dt, which means how the number of moles of reactants is changed with respect to the progress of the time, right? So as a reactant we understood, so we understand the rate of disappearance, right? The rate of disappearance, which means with the progress of the reaction, with the progress of the time, the reactants will get interact, reactants will get consumed, and reactants are disappear from the reaction environment due to, and will get converted into new products, right? So from the nature of the curve, we understand that as the time, as the progress of the reaction, as the progress of the time, the number of moles of reactants are decreased, right? The number of moles of reactants are decreased. So this nature of the curve is negative slope, right? So we'll have the negative slope, okay, negative slope, right? So let's say from up to certain time period, let's say we'll understand the nature of the curve. So we are dividing the curve into two parts. So in the first part, we are understanding the steeper slope, right? So up to, let's say, up to this much time period, from, let's say, from time t is equal to 0 to time, like from up to this time period, from 0 to here, right? From 0 to here. So where we can see the nature of the curve is very steep slope. We are having very steep slope in up to this much time period, right? So this steep slope indicates the fast, which indicates the higher rates of reactants, which means at the initial time period, where we have the higher concentration of species of which means we have the high concentration of reactant present at the initial time t, right? So which means if we are having the higher concentration of reactants present in the reaction environment, which means we'll have the higher rates of the reactants, right? Higher rate of the reactants, which means the speed of the reactants to consume and to convert into the product will be very high, right? So due to those reasons, we have the steepest slope in this particular time period. Okay, so which means at the initial time period where we have the higher concentration of A, we'll have the higher rates of reactants, which means at very high speed the reactants will get interact, they will get react, and they will get converted into the new products, right? So due to those reasons, with the higher concentration of reactants, we have the higher rate of the reaction, due to which we have the steepest slope, steepest nature of the curve. Okay, so at up to this particular time period, we have the higher rates of the reactions, and more number of reactant molecules will get converted into the new products. Okay, so due to which we have the negative slope, and afterwards in the second part, the slope in the second part, the curve flatters, right? The curve is flattened, right? So this flattening of the curve, which indicates the all the reactants are consumed. Okay, all the reactants are consumed in the reaction environment, which means the reaction is over, right? So there are no more reactants present in the reaction environment to carry out the reaction, which means, or we can also understand likewise, in this time period where we are having the flatter curve, which means we have the lower concentration of reactants present in the reaction environment. Okay, which means we are having the lower concentration of reactant molecules present in the reaction environment. Due to the lower concentration of reactant molecules, we have the lower rate of the reaction of reactants, right? We have the lower rates of the reactants, right? Which means due to the lower concentration of species of reactants, the rates of the reactions are very slow, which means the rates of the reaction, which means the speed of the reactants will be very slow, which means reactants will slowly interact, they will slowly react, and slowly they will convert into the product. Okay, and likewise the reaction will come to the completion. Okay, so I hope it is clear, right? So let's say at the time t is equal to 0, we have the 100 molecules, right? During the progress of the time, the reactant molecules will get consumed, right? So let's say, for example, at time t is equal to 5 minutes, first of all, at time t is equal to 0, we are having the 100 molecules. Okay, and during the progress of the reaction, the reactants are getting consumed. Okay, and after at time t is equal to 10 minutes, if we analyze the sample by using the different analytical methods, right, and we do the analysis of the sample, right, from which we are finding out the after time t is equal to 10 minutes, we have the 70 molecules of reactants present in the reaction environment, right, which means 30 molecules already consumed and transformed to the new products, right? And after, let's say, at time t is equal to 20 minutes, if we analyze the reaction sample, okay, and where we found we are remaining with the 20 number of molecules, reactant molecules, which means more reactants are consumed and they are converted into the new products, right? So likewise we can understand the nature of the curve. So as the reaction progress, as the reaction proceeds with respect to the specified time, the number of reactant molecules change, okay, and they will get consumed and will get transferred to the new products, okay. So as the reaction proceed, as the time proceed, we'll have the decrement in the reactant molecules, right? So I hope the first curve is understood, right? Now let's say in the second curve, which indicates the NB. NB represent the product, okay, product, okay. So how many number of moles of product is generated in the reaction environment during the progress of the reaction, right? So let's say at time t is equal to 0, okay, at time t is equal to 0, when we are feeding the reactant in the reaction environment. So no reaction, so we have no reaction, right? So reaction is not proceed, right? So reaction is not proceed, which means we have no product, right? So due to which NB is 0, no product is formed, due to which NB is 0, right? So here also we have two parts, right? Let's say in the first part, let's say from time t is equal to 0, where we have NB is equal to 0, and let's say up to this part, if we see up to from here to this part, okay, from here to this part, where we can see we have the steepest slope, okay, and the slope is in the increment, okay. So from this nature of the curve, we can see with the progress of the reaction, with the progress of the time, the new products are getting generated. And after some time period, the curve is flattened, which means now no more new products are formed, okay. So all the reactants are consumed and all the product is formed. So due to those reasons, this curve flattens after particular specified time period, right? I hope it is clear, okay. Now let's say here also we have two, okay. I request you to please make a note, okay. I request you to please make a note. I hope you have made the notes, right? So let's say now we'll understand this curve, okay. So let's say here also we have two parts, okay. So in the first part, say here in the first part, where you can see we have the steepest slope, right? So here this curve, the nature of the curve, which indicates is the progress of the reaction, is the progress of the time, the number of product molecules are generated, right? So here also we have two parts. So let's say from here at time t is equal to 0 and up to time t. So let's say this part up to here, from here to here, from here we can analyze that we have the steepest slope, right? We have the steepest slope, which means same way at the initial time period, right? At the initial time period, we have the higher concentration of the reactant species. Due to the higher concentration of reactant species in the reaction environment, we'll have the higher rates of the reaction, right? We'll have the higher rate of reaction, due to which the rate of reaction, which means the speed of reaction, right? We'll have the higher speed of reaction, due to which more the reactant molecules will get converted into the new products, right? So due to those reasons, we have the higher rates at this time period, right? So due to which more new reactant and more new products will be generated at a very higher rates, right? So at a very higher rate, the more new number of molecules of product will produce. So due to which in this part of curve we have the steepest slope, which indicates the dNB by dt, which means the change in number of molecules of product B with respect to the time, okay. I hope it is clear, right? So with the progress of the reaction, with the progress of the time, the more new number of molecules of product will generate, okay. With the higher rates, the number of moles of generation of the product will be very high, due to which we have the increment in the curve, right? And after some particular time period, let's say at here in this part, right, in this time period, where you can see The second part is in this time period where we can see the curve flattens, which means all the reactants are consumed and all the products are formed, due to which the curve flattens, right? Okay, I hope it is clear, right? So here in this figure we have the positive slope. Okay, why positive slope? Because with the progress of the time the number of moles of product is formed, okay, due to which we have the positive slope, right? I hope it is clear. Now let's say in order to derive the design equation, we are using with respect to the reactants A, right? So this differential form of mole balance can be written in the form of reactants, right? So let's derive. Okay, so let's say here Rj represent the rate of reaction of any j species with respect to the reaction volume is equal to the change in number of moles of j with respect to the time, right? So let's say Rj, which means here we are writing with respect to reactants, so minus Ra we are writing. Okay, why minus Ra we are writing? We already understand in the previous lectures. Okay, if you want to recall, I request you to please watch the previous lectures. Okay, so minus Ra represent the rate of disappearance of reactant A in the reaction environment, right? So minus Ra into V is equal to dNj by dt. Okay, so here we are writing minus Ra into V will be equal to dNa by dt. Okay, because we are deriving with respect to the reactant molecules, reactant species, right? So Na represent the number of moles of reactant species, right? So minus Ra into V is equal to dNa by dt, right? Now let's say if we want to find out like how much time, what time is necessary to reduce the initial moles of reactants from Na0, Na0 to Na1, okay, and from NB0 to NB1, right? What time is required, what time is necessary to reduce the initial moles from Na0 to Na1, right? So in order to calculate the time, we will make the subject as dt, right? So dt we are making the subject, and we can rearrange the above equation and we can write likewise dt is equal to dNa by minus Ra into V, right? So we are integrating the above equation, okay, and we are applying the limits. Okay, so limits are at time t is equal to 0, we have the Na is equal to Na0, right? So let's say at time t is equal to 0, we have Na is equal to Na0. Na here, so what does this means? Na is equal to Na0, which means number of moles of species A, which will be equal to initial amount of moles, right? And at time t is equal to t1, at time t is equal to t1, right, we have the number of moles of reactants will be equal to Na1, right? At time t is equal to t1, the number of moles of reactants will be equal to Na1, right? I hope it is clear, right? So limits are at time t is equal to 0, we have the Na0, and at time t is equal to t1, we have the Na1, right? So by applying these limits to the above equation, okay, so integration of dt, so we are integrating all the above equation, right? So we are applying integration to the both sides of the equations, right? So integration of dt, okay, integration of dt from the limit initial time to the final time t0 to t1 will be equal to integration of dNa by minus Ra into V from initial molar concentration, initial moles of Na0 to final mole Na1, right? Okay, and we already know the integral relations, right, of mathematical terms. Okay, how we integrate? So integration of dt is t, right, and the limits are from t0 to t1. Okay, so limits are from t1 to t0, right? So t1 minus t0, t0 is initial time, so t0 is 0, so we are remaining with the t1, right? So t1 is equal to the integration of dNa by minus Ra into V, and from the limit Na0 to Na1. So here we have derived the design equation of the batch reactor, right? Now we already know that Na1 is less than Na0, right? Obviously Na naught is greater. Okay, initial concentration, initial moles of reactants are higher, and after a certain time period we are remaining with the smaller concentration, smaller moles of the reactants, right? We are remaining with the less molecules of the reactants with the progress of the time. Okay, so Na1 is less than Na naught, right? And we already know the rate of the reaction A is less than 0. Okay, because A is consumed, and we are writing with respect to reactants, right? So what are the rates of reactants? What is the rate of disappearance, the rate of generation with respect to the products? We already discussed, I already explained you. If you want to recall, I request you to please watch the previous lectures. Okay, so let's say now, so this is the final design equation of the for the batch reactors, right? And we can also write in the other form as if we flip the limits, like if we flip the limits. Okay, so let's say if we flip the limits, so we have this form of equation where d1 is equal to integration. If we are flipping the limits, right? So from Na1 to Na, dna upon Ra into V, right? So we can write both the forms of design equation for the batch reactors, right? So here we have derived the design equation of batch reactors, right? And so what are the practical industrial uses of the batch reactors, right? So batch reactors can be applicable to the fine and specialty chemicals, right? In order to manufacture, in order to synthesize the fine chemicals or the specialty chemicals, we use the batch reactors, right? In the research and development, we are using the batch reactors for the kinetic studies. Okay, in order to study the reaction kinetics, in order to generate the rate laws, the rate constants, the data, we are using the batch reactors for the research, right? In the pharmaceuticals and the biotechnologies, right? In order to manufacture, to synthesize the pharmaceuticals, medicines, or the any products, right? A very pure product, right? We are using the batch reactors. We also have the glass line batch reactors, stainless steel batch reactors, jacketed batch reactors, right? Jacketed batch reactors are used for the transferring the heat to the reaction environment or removing heat from the reaction environment. Okay, so in the jacket where we flow the cold stream or the hot stream, and due to the heat transfer, the heat or the heat is transferred from the either from the jacket to the reaction environment, or the heat is removed from the reaction environment to the jacket to the fluid which is flowing in the jackets, right? So in order to start up new pilot plants, right, we use the batch reactors, right? In the food and beverages industries, we use the batch reactors. In petrochemicals and polymer industries, in cosmetics, and personal cares, in agro and fertilizers industries, right, we utilize the batch reactors, large vessel batch reactors we utilize, right? For the treatment of the water, in order to treat the water and to purify the wastewater, we use the batch reactors, right? And in the R&D and pyro plants, in the universities, in the research labs, we use the batch reactors in order to develop any process, in order to study in detail the kinetics of the reactions. We utilize the importance of the batch reactors. Okay, and in order to scale up, in order to study the scale up data, how we'll scale up the all the reaction kinetics or the all the reaction parameters. Okay, we try to use the batch reactors for the scaling of the studies, right? And for the understanding of the detailed reaction kinetics of the reactions, we utilize the batch reactors in the research, right? So I hope it is clear, right? So in today's lecture, we understand the how we are implementing the universal general mole balance equation, and we are deriving the design equation of the batch reactors, right? So I hope it is clear, and if you have any doubts, then feel free to comment below. I will try my best to reply to your comments. Okay, and I request you to please subscribe to my YouTube channel, please like the video, and please share this video with your friends. Okay, and until then, stay focused, keep learning, and stay curious. Thank you for watching my video. Jai Ambe.